So unfortunately, our next speaker can't be with, but she's Katherine Bryant. She's based at Aix-Marseille University, and this is going to be a bit of a shift to the kind of theme of the rest of the talks today, where we're really kind of moving forward with the idea of putting species in a common space, whether that be through diffusion, anatomy, structural, functional connectivity, or even evolution itself, and using trajectories of time, whether it be relatively short or the evolutionary lifespan itself. So with that, we're going to start with Katherine's talk, which she has recorded for us.
Hello, my name is Katja Hoyer, and I will be talking about phylogenetic.
That's Katja Hoyer. She's also doing a pre-recorded talk. There should be another video.
Comparative
No,
neuroanatomy.
not this one. No, there's.
Hi, everybody. I wanted to talk to you today about anatomically informed comparative neuroimaging. So understudied species can be a rich source of insight. So we have new model species that are being tested out and deployed in different clinical applications that are not as well studied, but also looking at species that just typically aren't studied by neuroimagers can give us access to new kinds of phylogenetic analyses to provide information about human brain specializations, human brain evolution, and brain evolution in general. Also, by looking at species that haven't been studied before, we can start to generate and test hypotheses about the relationship between brain and behavior and evolution, structure and function, how these are connected to the niche that an animal occupies. So neuroimaging in these understudied species does pose challenges. So the first main one is that software that's used for neuroimaging analysis is not designed for these species. It's usually designed for humans. Sometimes it's specialized for macaques. But outside of that, generally you don't find these software are not designed to handle them. So there'll be some things that we have to do to assess this and tackle it. Also, another problem is brain organization is highly variable. So across mammals, you have a wide range of variability in sulcal organization and just the gross anatomy of the brain that will cause you challenges as well. And a lot of us aren't biologists or didn't get biology training. And so some of this might seem a little daunting at the beginning. So I'm going to go over an approach to handling these problems. First, I'll talk about where. So how do we identify homologous areas in less well-studied species? I'm going to talk about when to move from manual interventions on analyzing these data to automated approaches. And I'll talk about what kinds of analyses you can do once you have your data processed. So one thing that will be very critical is anatomical resources. And if you look in the literature, sometimes you can find historically species that aren't typically studied were studied by people like Brodmann, Bailey and von Boone, and other early neuroanatomists that created resources. So these are invaluable if you find them, because generally they used in a landmark approach, they looked for homologies and they labeled them as such. Information from blunt dissections from this era are also very helpful. More recently, there's, of course, work in track tracing and histology that sometimes has been done on less well-studied species, so it's worth tracking down that as well if you can find it for the species that you're working with. And then more recently, we are seeing more and more neuroimaging-based atlases of typically not studied species. And of course, any kind of scan that you have, even if it's not an atlas, you can look at scan information that can help you as well. So another thing to keep in mind is what can be more challenging or less challenging when it comes to the species that you've chosen. So if you pick a species that's better studied, of course, that will be easier. If it's a species that's related to species that are better studied, it will be easier. So if you have an old world monkey that's a similar size to a macaque, you may not have to do too much work to identify homologous landmarks. If you have a number of scans of different individuals and you can build a template, that's going to improve that's going to help this process as well because you'll just have a nicer template scan with less individual idiosyncrasies. Postmortem scans are very good for this kind of analysis because you can have much longer scan times It can boost your SNR, and you can have a better quality scan that can help with this. And then, of course, brains that are larger will be a little bit easier than smaller brains that have fewer landmarks, fewer sulci. Those can pose a challenge. So, like I said earlier, homologous areas were sometimes already identified through cytoarchitectonics from earlier studies, more traditional studies. So here's a chimpanzee brain, which essentially they used very similar landmarks that had been done in humans and other species. So there you have your homologies already laid out for you, which is always very nice. Blood dissection information, of course, is very useful as well. A lot of this data is basically was presented through these like pen and ink drawings, but they can be really invaluable anatomical resources for showing you where tracks are moving. And then, of course, track tracing, like Schmahmann and Pandya, for example, and their excellent fiber pathways of primate brains. This information gives you direct homologous information for white matter pathways that are the same, that are similar to humans, but they obviously have differences in terms of how they're organized in macaques. And also, if you have if you do use myelin mapping with your scan data, this is incredibly useful because the higher concentrations of myelin are focused around primary areas. So if you're not sure where a primary area is, or you need to identify something near the primary area, this is wonderful data to have. You can also look at homologous areas using FA maps. So if you have an FA map of your species and then other species that are somewhat related to your species, you can then start to break down where are these tracks based on these spatial, their spatial orientation, using the sulci externally to guide you where you search for them. This can be helpful. Here's an example of something that would be more challenging. So a Saki monkey, it's not been studied. It has very few sulci, so it's hard. It can be challenging to orient yourself anatomically, and it's only one individual. But we can see it has a nice FA map. So already the FA map can give us information about where the tracks are, and then once we identify the tracks, we can move forward from there. So how about manual to automated? When to do what? So this is everything from tweaking things during pre-processing, and looking at creating region of interest maps, all the way to downstream analyses, essentially. So template generation, segmentation, doing tractography, and something Sean will talk about later, I won't talk about, is the common space approach. So now we have our, we're going to start pre-processing. So what do we have to do? Well, first of all, you need to check when you do your very earliest parts of the pipeline, for example, brain extraction and tissue segmentation. So you can use automated approaches for this, but you need to check to make sure that they have been executed correctly. And the reason is because these brains are different sizes, they're different globally in terms of like their shape. They're not well suited to these programs. So you need to go in and manually correct them, or at least verify that they've been done correctly. And the same thing for the tissue segmentation. If you have a number of species and you want to a number of individuals of your species and you want to create a template, you could end up with something like this. This is this is work that our lab is doing, creating templates of neonate baboons. So we spent time on each individual, getting the segmentation corrected, getting the brain extraction corrected, and then we're able to successfully create a template. Okay, so if you're interested in tractography and you now have your anatomical landmarks laid out, you've done your pre-processing, so now you can use these anatomical landmarks to create your tractography recipes. So this is an approach that I use. So rather than doing an automated tractography or cluster analysis, I like to be able to directly pull out the tracks that we know are homologous and that exist in humans and exist in macaques in other primate species. So I do this by creating a series of recipes in order to extract out these different tracks. Then we could do them across species. We just have to identify the homologous landmarks, so we're always putting our region of interest mask in the correct location. And this, you can also, if you are creating your own custom set of recipes to do these kinds of virtual dissections, you can use extract, which is a part of FSL's package. And Sean Warrington will talk about this as well. But it's a way sort of like you can plug in what you've done and then it gets deployed through extract in order to do the tractography and organize your results. So that's very nice. Okay, how about what kinds of analyses we can do? So we can do everything from a species level analysis. So maybe you have a species and you just want to create a map of it, or you want to create an atlas based on several individuals of that species, that's something that you can do. You can do a comparative analysis. So we can look at differences between species, like you compare two different species and look at those differences. And finally, if you look at more than two species, you can do what's called phylogenetic inference and start to delve into how evolution may have shaped brains. So in terms of maps and atlases, this is a recent example of a Lar gibbon that I did. And so we actually started out, we spent a lot of time making sure that we knew what the sulci were, because there was conflicting accounts from older literature that we had to sort through, compared to its closest related species that we had agreed upon anatomical terminology for, and moved from there. So we started with that. Then I was able to create my recipes. I was able to extract out all the white matter tracks that I wanted to, and publish it as a white matter map. And then the nice thing is now these recipes are available online. So if somebody wants to go ahead and say, I have a series of gibbons that I would like to analyze, you don't have to start from scratch. That work has already been done. So how about you want to compare two species with the same analysis? So one thing you can do is once you have so again, I am focusing a lot on diffusion tractography because that's what I would do, but here's an example of how that might work. So say you have your white matter atlas, and you also have mapped out how the white matter is projecting to the surface of the brain. So now you have like blueprints of like the relationship between the tract and the surface of the brain. You can then do a matrix-based approach, for example, KL divergence, to look at the differences between the two species in terms of how they map out their different white matter tracts to the surface. And you can end up with something like this. So these hotspots are telling us that in these regions of cortex in the human above, they have fingerprints of how the tracks are reaching that area that don't match well anything in the chimpanzee brain. So in that way, we can say, okay, these are differences between these two species. And we can even interrogate that further and look at what's driving those differences. So we can say, well, it looks like humans evolved this, or we have this specialization in this area. So how about phylogenetic inference? So phylogenetic inference, so if you're not an evolutionary biologist, I'll briefly just say, frequently we use something called the maximum parsimony approach. And so essentially, if you have two possible trees to describe how a phenotype changed over time, so like different state changes, usually the best educated guess is that the tree with the least amount of changes is probably the most likely to have occurred in evolution. So we can use this maximum parsimony approach and apply it to these kinds of white matter maps. So here I'm just giving you an example of some of my work where now that I have maps for all these species, I can then start to break down how many state changes would be the least amount that would result in the results that I have observed. And so in this example, I basically found that looking at all the tracks across the brain, it really is the arcuate and also the ILF that seem to have changed across species, and I can even start to build out, well, when did the arcuate changes occur, and to which common ancestor? And then I'm also kind of dabbling in ways to also present this visually in terms of what are the changes, can I do a reconstruction of what the most recent common ancestor would look like? I want to briefly talk about data repositories because they're really an amazing resource for this kind of work. So a good example is the University of Oxford's WIN Digital Brain Bank. Within there, they have the Digital Brain Zoo. So this is a series of data sets on less well-studied species that you can request to download and use yourself. And these are post-mortem scans that were done over a long period of time. So they're very nice data. And another thing that you can do is say you're working with these kind of data and then you're producing maps, you're producing atlases, you're producing other kinds of resources. You can make those available as well on any repository that you'd like, and then other people can have access to it. And then that just builds out this sort of like evolutionary knowledge. So the more people do it, the more they share it, the more other people can use it for their comparative analyses. I think it's very exciting. So in summary, we live in exciting times. We're getting a lot of high quality postmortem imaging of understudied species, and people are sharing this information. So not just the scans, but as people are creating atlas resources, they're sharing those as well. But you should keep in mind, if you're doing this kind of work, that mapping understudied brains does require anatomical knowledge. So you will have to delve into that a bit. You can't assume that the software is going to handle the brain properly. Don't be afraid to do some manual corrections. I really strongly recommend it. And if you're going to start doing phylogenetic analysis, you're going to need to delve a little bit into evolutionary theory. I think it's worth it. I think it's exciting and interesting, and I'm kind of looking forward to in the future new approaches for kind of combining phylogenetic approaches and neuroanatomy approaches. So with that, I wanted to thank you all for your attention, thank my collaborators and my funders.